In addition to the action potential, neurotransmitters or neurochemicals are the primary way that the brain works, or signals, processes information. So we will now cover all of the major neurotransmitter systems, some other neuromodulators that are involved, and then try to tie a lot of these concepts back together in an applied fashion in the next PowerPoint by exploring epilepsy and seizures and how these things can interact. But for this particular PowerPoint, I would like you to be able to identify all major neurotransmitter systems covered in this presentation, including corresponding specific receptors by name and whether they are ionotropic or metabotropic. I would like you to be able to identify the origins of acetylcholine, dopamine, serotonin, and noradrenaline pathways. I would like you to be able to compare and contrast the primary amino acid neurotransmitters. And I would like you to be able to identify the peptides and gases covered in class and now how they act as neural signaling molecules. So I've already brought up Henry Dale, so let's start there. Henry Dale, did so many phenomenal things. I mean, this idea that like the Dale's law was wrong. I mean, Henry Dale, discovered the first neurotransmitter. It was part of the discovery. Technically acetylcholine had already been synthesized, but it was the first recognized neurotransmitter. Dale, along with Lowy, shared the Nobel Prize for it, and it was, considered to be the first neurotransmitter. It was secreted from the vagal nerve. That was the first discovery of it. And, most neurotransmitters in the brain weren't actually discovered in the brain. They were mainly discovered in the periphery, many of them in the gut, actually. And again, it's very popular now, this idea that the microbiome interacting with the brain, and most certainly it does, as do most, peripheral organ systems. The brain is the integrator of what's going on throughout the body. And really, not only what's going on in the gut, many of these things can affect our emotionality and behavior. But it is all still integrated by the brain, which controls the behavior and the emotion. So acetylcholine was mapped by really the enzymes involved in its synthesis, and it was also one of the first neurotransmitters to be, described structurally as a result. The cholinergic nerve cell bodies and projections contain the neurotransmitter acetylcholine. There are two main acetylcholine pathways in the brain. One arising from what's known as the PPT, or peduncular pontine tegmentum, or nucleus. The other arising from the basal forebrain. The nucleus basalis of Meinert is very important in the field of Alzheimer's disease research. Alzheimer's has had a bit of a roller coaster of, an understanding over the last couple of decades. As you probably learned in 2220, or just Alzheimer's is commonly associated with plaques and tangles. And recently, a big joint effort to try to find a treatment for Alzheimer's disease was able to target those and break them down in animals. And then they brought this technology to people, the pharmacotherapy, and it didn't treat Alzheimer's. I don't know that it's so clear as to whether or not it just wasn't breaking down the plaques and tangles in adults, in humans, like it did in animals, or if the plaques and tangles are really a bit more of an epiphenomenon, sort of like something that occurs over the course of the disease, but isn't necessarily the causal domino. Two causal dominoes that I think might be important there. One is just mitochondrial dysfunction across most neurodegenerative diseases, including mitochondria, but even traumatic brain injury. You see disruptions in mitochondrial function. So that could be a very common denominator across neurodegeneration. One of the first things to go is the mitochondria, and how you target that and fix that, I don't know, that's going to be a problem. But it could be, a potential target. And if you know the problem, then you can start looking for fixes. The other might be cholinergic degeneration in the nucleus basalis of Meinert. Very early on in the disease, it's thought that cholinergic neurons that innervate the cortex degenerate, and their degeneration might be one of the initial dominoes in the cascade of events that leads to cognitive decline. So that would be one very important potential role of acetylcholine neurons of the nucleus basalis of Meinert in the basal forebrain. There are two types of acetylcholine receptors, nicotinic and muscarinic. For nicotinic, most are ionotropic and excitatory. Muscles in the neuromuscular junction use nicotinic acetylcholine receptors. Paralysis can be induced by an antagonist of the nicotinic receptors, such as curare. It is commonly stated that nicotinic receptors are in the periphery and muscarinic receptors are in the central nervous system, the brain. That isn't necessarily true at all. There are nicotinic receptors in the brain, and I've seen that they are actually capable of depolarizing dopamine neurons very quickly. I've seen this myself. This is probably occurring at the level of the brain. So, while definitely nicotinic receptors are important in the neuromuscular junction and abundant in the periphery, they do exist in the brain as well. Muscarinic receptors are metabotropic, meaning that they would need to activate a G protein receptor. Muscarinic receptors are typically slower than ionotropic receptors. And they, in this case, can be either excitatory or inhibitory. This would be determined by the G protein complex associated with the G protein or the muscarinic receptor. If it's coupled with the GS form of the G protein complex, it would be excitatory or stimulatory. Think GS stimulatory. The other would be GI. GI, if it were coupled to the muscarinic receptor, would be inhibitory. So GS stimulatory, GI inhibitory. Just one very common way to know this. GO receptors tend to be like GI receptors, also inhibitory. There are also these things called GQ units, but let's not go there. It won't be necessary for the test. Muscarinic acetylcholine receptors can be blocked by atropine or scopolamine to produce changes in cognition. Again, in going back to this idea that cholinergic function, it has many functions, but it does also seem to have a role in cognitive performance, in addition to wakefulness, many other things. But, that role in cognition could be compromised with disruption of the nucleus basalis of Meinert. The modulatory monoamine neurotransmitters are some of the best, most popular amongst the general public because we commonly target these for a variety of psychiatric disorders. Many of them contain slow-acting metabotropic receptors that would produce more subtle changes than you might get by mucking around with GABA and glutamate ion channels. So there are two main classes of monoamine neurotransmitters, the catecholamines and the indolamines. This would be really based off of organic chemical structure. The catecholamines are dopamine, epinephrine, and norepinephrine, and the indolamine, it would be serotonin. And the catecholamines also all share a similar synthesis pathway. Dopamine is found in neurons in the nigrostriatal pathway, which originates in the midbrain, specifically the substantia nigra, and innervates the striatum. This pathway is important in motor control, and neuronal loss is a cause of Parkinson's disease, as we've already discussed. The mesocorticolimbic or mesolimbicocortical, as I think your book uses, and there are slightly different names for these pathways. Most of them include the key anatomical elements here. But anyway, the mesocorticolimbic dopamine pathway originates in the midbrain in the ventral tegmental area and projects to the limbic system and cortex. Dopamine in this pathway is involved in goal-directed action, reward, reinforcement learning, and there are also abnormalities that are thought to be associated with positive symptoms of schizophrenia, although that part is still somewhat controversial. Here are the two pathways, the mesocorticolimbic or mesolimbicocortical, as your book says, and the mesostriatal or nigrostriatal dopamine pathways. You can see that the nigrostriatal is somewhat dorsal to the mesocorticolimbic pathway. And also in regard to function, think of these things going back to the idea of cortical subcortical loops. One sort of leading idea here is that, sort of early on in learning a new behavior, this would be primarily orchestrated by the more ventral mesocortical limbic dopamine neurons. So these would be capturing or encoding information about the value of a new outcome. And then over time, the pursuit of that outcome becomes more automated, more automatic. Similarly to playing the piano, early on, you might be, relying more on that mesocortical limbic dopamine pathway as you're sort of learning the new procedural memory. And then over time, it becomes more automated. Similarly with sports, like a golf swing, very early on, probably you see this sort of mesocortical limbic encoding of what's going on, and then sort of a ventral to dorsal gradient as the task becomes more automated. Norepinephrine or noradrenaline is released from the locus coeruleus of the brain and projects widely throughout the brain. Norepinephrine is also known as noradrenaline, and its cells that produce it are noradrenergic. There are 4 types of norepinephrine receptors, all of which are metabotropic. This would be the alpha-1, the alpha-2, the beta-1, and the beta-2 noradrenergic receptors. Similarly, there are 2 dopamine receptors, the dopamine 1 and dopamine 2 subtypes, both of which are metabotropic. The norepinephrine system modulates processes that are widespread, but these include generally mood, arousal, and sexual behavior. Here you can see the noradrenergic neural network in the brain. You only have one site of origin to memorize here, the locus coeruleus, which is nice, and this would project widely throughout the brain. Serotonin cell bodies are mainly found in the raphe nuclei of the brain. And again, their serotonin fibers project widely, very common amongst monoamine transmitters. All of these are pretty have widespread action through the brain. Serotonin is implicated in sleep, mood, sexual behavior, and anxiety. Antidepressants such as Prozac increase serotonin activity with effects depending on what subtypes are affected. We'll go into more of this later, but the pharmacotherapeutic effects of SSRIs, like Prozac, in treating anxiety and depression are thought to involve a sort of a 2-week period of changes in the serotonergic system. And to some extent the noradrenergic system too. And in fact, the tricyclic antidepressants and these sorts of things have probably more effect on the noradrenergic system in producing positive effects in the treatment of anxiety and depression. But, in the case of SSRIs, there might be more selective effects on the serotonin system, and this would involve things like downregulation of select postsynaptic receptors of the serotonergic system, of which there are many, and possibly changes in serotonin synthesis and autoreceptor regulation, that 5-HT1A receptor I was alluding to before, which is presynaptic and tends to inhibit serotonin release. The idea is that it takes 2 or 3 weeks for these SSRIs to have positive effects in probably around 50% of the population that are depressed. And the idea is that the benefit doesn't come from increasing serotonin levels as much as it does from changing long-term changes in receptor expression. In fact, it might be quite the opposite of this sort of the idea that maybe the person has abnormally high or sort of peaks of serotonin at certain times. It's not like they just don't have serotonin. But the idea is that maybe the postsynaptic response is super sensitive to these sort of peaks of serotonin that come on in an anxiety-inducing event. And so with the SSRI, what you're doing is sort of hammering the system. You're desensitizing the system. You're making that postsynaptic response less sensitive, and you're also probably reducing the presynaptic responses. So just like you have a single site of origin for the noradrenergic system in the brain, the locus coeruleus, we're only going to learn one site of origin for the serotonergic systems in the brain, the raphe nuclei. And of these, there are dorsal and ventral portions of the raphe nuclei that really are pretty distinct. But for the purpose of the test, just know that the raphe nuclei would be the site of origin of the primary serotonergic pathways in the brain. The brain also contains two primary amino acid-based neurotransmitters. These are glutamate and GABA. Glutamate is the primary excitatory neurotransmitter in the adult brain. This is primarily determined by its receptors. There are three ion channels of the glutamate system: AMPA, kainate, and NMDA receptors. Many people forget about the kainate receptors. I would not if I were you. There are also metabotropic receptors that glutamate binds to. I'm familiar with types 1 through 7. There's some excitatory and some inhibitory forms. Again, this would depend on the effector, that secondary effector, the G protein that is coupled to the metabotropic receptor. Is it GI inhibitory, GS stimulatory, excitatory? mGluR types 2 and 3 tend to be coupled to the GI or inhibitory subunit, and I know that types 1 and 5 tend to couple with the GS subunit by contrast. These are all involved in excitotoxicity, which we've already learned about a little bit as well. Glutamate-induced excitotoxicity, which also is very much associated with NMDA ion channel as well, and connexins and gap junctions, as we learned about. But excitotoxicity would be neural injury, such as stroke, that may cause excess release of glutamate, which was toxic in neurons in large concentrations. Astrocytes are also involved in the uptake of glutamate from synapses. These glia tend to be positioned right outside the synaptic clefts and have the machinery to take up and recycle excess neurotransmitter and can even signal some on their own. GABA is a primary inhibitory neurotransmitter of the brain. There are ionotropic GABA receptors, with GABA A being the primary example here. This would produce fast inhibitory effects. It was what barbiturates would directly act on. Benzodiazepines like Xanax and Valium are allosteric modulators of this ion channel, meaning what they do is they don't actually activate it enough to allow inhibitory chloride ions to flow through it. But what they do is they produce a conformational shift so that when GABA binds, it's more effective at inducing chloride ion influx. GABA B receptors are, by contrast, slow, metabotropic. These still tend to be coupled to GI, GO subunits and would be inhibitory in nature. One example of a GABA B agonist is baclofen, which would be used to reduce muscle spasticity. It's also been shown to treat alcoholism, interestingly. Alcohol has a, really it's probably the most embarrassing thing about neuropharmacology is we don't completely understand how alcohol, the most commonly abused drug in Western culture, acts on the brain. How it seems to be probably widespread and complicated, but it certainly has actions on the GABA system, but it's not only a GABA agonist, and it's certainly not clear-cut. The brain is complex, guys. It's not just all about classic neurotransmitters, and there's really just not one neurotransmitter chemical in one cell that signals. These things are much more complex and nuanced than, we even understand at this point in time. But peptides can act as neurotransmitters at some synapses. Opioid peptides like beta endorphins and enkephalin dynorphins would be a good example. Beta endorphins really are what opioid drugs mimic. This would be the prototypical agonist at the mu opioid receptor. By contrast, dynorphin is the prototypical agonist at the kappa opioid receptor. This is sort of the opposite of the mu opioid receptor, just as a very simple way of putting it. So if beta endorphin or morphine were to produce analgesia and euphoria by activating the mu opioid receptor, when you activate the kappa opioid receptor, this is actually used as a model of stress in animal models. Dynorphin would do this, and it's thought to be associated with drug withdrawal. And interestingly, salvia, salvinorin A, is an exogenous agonist for that kappa opioid receptor, the dynorphin, the neuropeptide binds to. And I've always found it very curious if people, choose to abuse this drug that is used to induce stress in animal models. It does produce a dissociative hallucinatory state at high dose, similar, maybe somewhat more comparable to what you would get with like ketamine or an NMDA receptor antagonist. But, why? Salvinorin A, I've never really understood the, abuse situation on that one. In addition, there are lipid and gas molecules. One very common lipid molecule would be the endocannabinoids. Interestingly, these are made on demand during periods of high neural activity. A lot of calcium influx leads to the on-demand synthesis of endocannabinoids that are made postsynaptically that then signal presynaptically across that synaptic cleft. Being lipids, they might even just diffuse through the plasma membrane and then activate presynaptic CB1 receptors, which tend to be coupled to GIGO receptor, and then would inhibit presynaptic release. And they're most commonly expressed on GABA presynaptic terminals, so this would lead to an amplification of the postsynaptic response. Similarly, nitric oxide is a gas that can be released from neurotransmitters and seems to act very similarly to endocannabinoids, the lipid signaling molecules, to retrogradely activate the presynaptic neuron to alter its release and then sort of indirectly activate the postsynaptic neuron. I tend to simply think of these as amplifiers. It's frankly a little more complicated than that. But the idea is that, maybe during prolonged periods of activity, in certain cases, you want to amplify the signal even further. And, one way to do that would be to release gas like nitric oxide. The other would be to synthesize lipids like endocannabinoids. So, we have electrical transmissions that can be even direct. We have classical chemical neurotransmitter transmission, but that is also modulated by neuropeptides, gases, and lipids. And here's a table that I think is a good summary. If you know this table, you should be pretty well prepared for the test for this unit. There's a lot of rote memorization here, but, if you don't know this, you're going to be somewhat lost as we, move in and really get into application in this. And, these are just sort of building block concepts. So here I need you to know, all of these various neurotransmitters. I want you to also know the second column, the known receptor subtypes, and then not as much in the function column, knowing a few things that I pointed out in the PowerPoints isn't going to hurt you, but I also want you to know the origin of these neurotransmitter systems, the ones that I covered in the PowerPoint presentation. The only thing here that I see might be a little different than what I said is dopamine receptors, known receptor subtypes. Here, I just want to clarify that there are indeed two major dopamine receptor classes. These are the D1 and D2 like receptors. D1-like refers to D1 and D5 generally. D2-like refers to the others. D1-like are typically coupled to the GS subunit, so they're excitatory. D2-like are coupled to the inhibitory subunit, so they are inhibitory.